those who are online also able to listen, right? Yeah. So mid-sem is though postponed, but the syllabus is going to remain same. So no need to worry. Yeah, but according to me, maybe that is a semester is going on. Maybe it's October to November, right? So there is maybe a chance in October or so maybe in a September end, they may have the semester mid-sem 2. In October, they are going to have the mid-sem 3. And then the final. 2 mid-sem. 1 and 2 or 3? Okay, so mid-sem 1, mid-sem 2, not a mid-sem 3. So maybe it is in our September, October, they may plan. It was in my mind that there are the 3 mid-sems are there. Whatever it is there. That is far better that you may have the 1 or 2, but I'm not going to increase the syllabus. Whatever been told earlier, it is going to remain same. So although we have about to complete, okay, that point is already covered. What just I had told you people, just one point is just left. After taking this topic, I'm going to take you for that because they have given in 2.1, that is in first line they have given, but it must be taught to you people after completing some of the basic points. Okay, sometimes while writing the syllabus also, they may have in their mind to teach or give some of the particular targeted part. So while writing, maybe it is in a first line or second line, this may have come. But while teaching, one should able to understand first basic parameters. What are they? Then if I'm going to take you for the topic, it is like that, like effect of pH, temperature, and other all our aspects are covered into it. So if you're not knowing about the pure culture or the colonic characteristics or the effect of certain parameters or the chemical or nutrient, how you can able to understand on the first point? Okay, so basic things are there that I'm looking always to take one by one. Which point is very much essential for you people to know and understand, that I will take first. Okay, either of the syllabus is lying here and there, because while preparing the syllabus is always happens. Okay, so they have given just one paragraph, nothing else. In all the places they have given the one paragraph, just one comma or something dots are there. So they are looking to make you understand at least few selected points are required to get understood. Okay, same paper name is there or book's name is there. Maybe you are going to see in the another place, in the another university or another place, you will find that they have given the many topics, or may have the less than that. But they may are going in a superficial, or they may are going in a detail, because that parameter is not been shown over here. Okay, so always if you are looking to do some comparisons, okay, it is a good, but while doing the comparisons, the level of the girls, how much they are taking or what you are learning is going to change. Sometimes like microbial growth, okay, in your syllabus is the 1 unit. If you are looking to in certain university, maybe they have given is the 1 point. Okay, part of the 1 unit, just 1 point will be given. So you will say, you will surprise, what is this? You are learning for 1 unit and there is a part of 1 unit. So they are just going to learn about what is microbial growth. Maybe few points they are going to learn. But in your part, it has been given in a way that microbial growth, that means what are the basic elemental parts are being associated with the various kind of microbes. Okay, when you are looking to understand about it, that's the point wise is going on. So always it is. In your syllabus, some of the points are given in the different pattern. In the another, maybe you are working or learning, they may be given in different formats. And the same, it is like that. Maybe you are knowing about the credit-based transfer of the students transfer scheme. You are knowing, like from the one university, if you are learning in here, you can apply for the at least about 1 semester for the selected paper in another country also. In bachelor's, people are not majority are going. For the masters, they are going to. Even in PhDs, people are doing. So during the masters, even in a bachelor's, if you have learned certain points, so it is called the credit transfer. So one of them, you may have learned from the any university from the US or the Canada. The mark sheet will be offered from the Marwadi. Exams and everything was conducted. Everything was mentioned in that way that from the certain university like Penn State. Okay, so it is a credit transfer. So there the comparison of the syllabus is done. Maybe the same paper is offered in the fourth semester, not in the third semester. So what are the points are there that you are going to learn over there? That is the selections will be there. Committee will be going to decide about it. There they are going to ask to the faculties that what is the level of a girl or the level of the syllabus you are taking in a detail. So there we are going to facilitate to the your applied institute. That our agenda and the points are there. Till this we are going to describe or that in detail we are going to do topic is there. What they are going to say that our syllabus and pattern are telling we are going to in this much detail. So they are going to for a time period they are going to give the new syllabus for a while for the one semester. The topics may be not covered or may be covered according to their agenda and our agenda. Okay, new syllabus for a while it will be given to you. So maybe they are having some new pattern name and everything will be there. So such thing is there that people are looking to learn the cross country wise. The culture difference may be there. The learning behaviors, practical aspects are there. You may be knowing that this paper is taught by some of the prominent professor in the so and so places, and you are looking to learn from him or her only. So you can have that facility. So such kind of thing is going on. Maybe you are knowing or not knowing. I'm not so sure, but there exactly the syllabus level is required to understand. Maybe you are going in after bachelor's to the master's. Some of the time, they are going to give a bridge course. Maybe you are looking not for the master's, you may going for the PhD. Okay, or otherwise, they may provide a direct research degrees, MRes. Here it is a MPhil. So in MRes, what they are going to do, they are asking that few papers you have to learn before going in the research field. So there the comparisons will be asked for the syllabus. Whether our syllabus is comparatively according to experimental part or not. It is a microbial experimental part. Okay. So our major part is related to theory covered as well as the practical aspect is included. So it is related to the research or even. So if you may go for the MSc or you may go for the MRes. So when you are going for the MRes, that means you are looking to go in a research field itself. So they may give about MRes may go for the 1 or 2 years, 3 years, depend upon the way up to till you are going to complete your syllabus. Okay, that is a research targeted part you have to complete, the dissertation part. Before that, you have to complete their syllabus. You have to learn it. You have to clear that. So what they are going to offer, you may show your syllabus that already I have learned this topic. They are going to give their criteria. So it is like a join A and join B. Column A and B is there. If they are going to fulfill all their criteria, you can directly move for the MRes. Otherwise, you have to learn that theoretical part. You have to clear that credit. Okay, it may be a 4 credits, 3 credits, or 2 credits may be offered to you. Okay, that 2 credits, if you are going to complete, then the enrollment is allowed for the MRes. Otherwise, you have to complete again and again. Special class that you have to attend with maybe a bachelor with the master students during that time. The timetable will be there with you. It is like that. Currently, you are having the my paper, right? Because some of the students not able to because of certain conditions by accidental chance. He may be failed in one paper. Okay. So what he is going to do, he is going to come in a class for only one lecture. Then he may move. Like that only, you are going for the selective lectures, right? So then you are going in bachelor's or master's depend. The real comparisons of the syllabus start there. That is why people are choosing the several different universities according to their own wish, right? Why the people are like in a MSc. If you are just talking about the Gujarat, okay, there are the Ahmedabad, there are the Gujarat University, Baroda universities, several universities are there. Here it is the Rajkot, Saurashtra University is also there. This I'm telling about the state university. Private universities are also lying a lot number. About in Gujarat itself, near about 40 to 50 universities are there. About 40 universities are offering bachelors in microbiology. If you are going to do a comparisons for the all, it will be a different. So now If you're looking for joining any industries, what will be their criteria? To whom to select, why to select, and how to select. Okay, so their agenda will be fixed. They want special guys, maybe a technical expert, maybe a subject expert. Depend upon the time dependency, it may change. So they may ask for the sometimes the syllabus ask. What is their syllabus? What they have learned? Otherwise, they may start to ask from their results and mark sheets. What paper you have learned? Sometimes I may just ask like that only, right? Or many people are, what is the paper name? Okay, maybe you are right now in a sixth semester. Okay, what you have learned in the first semester? Okay, microbial physiology. Can you say that what sort of the topics you have learned? Maybe I'm just interested into a production. Okay, in my lab, I'm looking for the take two guys, those who are able to do a good production. Okay, fermentation tank is there. Then the growth curve will be a most essential component over here. If I'm going to ask the microbial physiology, and you just forget that unit name, microbial nutrients, then there's some photosynthesis, then we have learned some characteristics. You just forget the microbial growth. Okay, sure, what is gone. Then you are going to say that, wow, interview was very nice, but I don't know what happened later stage. I was about to get selected, but never been happened. Because their agenda is clear. Okay, if you're looking to do same thing, yeah, if you're looking for the masters in a foreign countries, then the interview online is being conducted for the research. They are asking for the SOP. Do you know about the SOP? Okay, statement of purpose. Why you are looking to come here. Okay, so what is your mentality? What is your target? What is your aim? What is your objective? Is the learning or the getting earned? Okay, then after they may have the interview panels. 4 or 5 people are sitting, maybe it's the online or offline, could be possible. Then they will going to ask the certain questions. Within a 10-minute of brief period, they will assess you what you are and where you are really standing. This is so there it will be every sort of the things will be because you have already applied. You have sent your transcript, right? Everything is with them. They are also asking for the syllabus copy, right? Many institutions are asking for the syllabus copy. So everything is within them. Within 1 hour, they are that much prudent that going to check it out that what could be your level. They are just going to look. Within a 10 minutes of period, in interview, they are going to say yes or no. So they are that much clever enough, understanding the phenomena about each person's. Then you are starting with a good morning and good afternoon as a all and all. They are not much looking about it. Yeah, that is good as a part of culture. Okay, but the second part is what you are, what is your motto to come here. And in reality, if you are knowing certain basic phenomena, it is not always that everything you are knowing. Maybe that time you are not able to recall. You can say that really I don't know. Okay, so whether you are going to stick on your opinion or not, or what you have told, it is there or not, or you are just managing to get engaged. Okay, it is able to understand by one. Many times I'm telling, sometimes I also do mistakes. I apologize. Or I may say that, okay, yesterday I have told, by chance, during that time I have told, but it was wrong. But right thing is what? Right. Because or it may be, over that time it is correct. Science is always a dynamic. One laws or the second laws are coming, or the experimental proof are coming, the theories are get falsified. So what is being taught today, it may be a totally wrong. But on that time, it was a correct thing. So it's a very dynamic. We have to live in the real current era. It is like the corona conditions has came. We have to learn in the online. People may not like to teach in that format, but the dynamic era make you push to do this. And this is the reality. So how much you are going to? So whenever it is been asked some of the questions, okay, you may start to give the answer. It may be a bluff. Okay, the persons before you are already knowing, and they will allow you to do a bluff. Oh, yes, it might be. It's going to happen like that only. Yes, you are right. Can you say further? So you will say that, okay, I able to manage. Okay, but already that fellow is knowing that you are bluffing. Okay, and then you are going to say that I have told everything. That stupid guys who are sitting there not knowing anything. They are just asking the few questions. I have replied a lot thing. Okay, so it is a very good negative sign for you yourself that you have created an impression that you are just bluffing. Okay, and this some of the times, the professors and teachers are not going to show any sign to before you that you are bluffing also. They're already knowing. They're going to keep in their diaries such things. That such guys are always do a bluffing with a smiling face. Professors also having the smiling face. And those who are not bluffing, those who are going to do very good things, just one mistake, they get punished. So they're telling, oh my God, I am always get punished. I am always get talked by them. Okay, then the bluffers are there. The teacher is not telling anything because the teacher want to make some changes in their students. Those who are bluffers, they are not interested at all to say anything. Okay, they are not caring for them. So whenever for the masters of PhD, okay, the students have been selected. What will be the behavior when the cultural programs are going on? So many from the country-wise, the globally, the persons are coming. Okay, whether he or she will be able to settle in my lab or not. If just a bluffing is done, some experimentations may going on, something may happen. Who will be responsible? So I don't like that, guys. Or else I may like such guys, so I will do such experimentations only. Okay. So what you are, where you are, you can say, and you can save yourself. It is nothing but the cheating with yourself. It is a really good say that cheating with yourself that I am able to manage many things. Right. Nobody has noticed, but it is unnoticedly noticed. Notice been informed to you any time, and you are saying that, wow, who I am. Okay, just get proud, get cheated with yourself, and then after you will realize that I will normal, nowhere. Those who are going to improve it, that get settled very well. Right. They are starting to settle very well. They got the things phenomena. They are looking to come here for the learning purpose some other time. Yes, college life, everything may happen. Once maybe it is good. One sometimes may go for the bunking even. Management will start to ask many things, where you were, how you were. That is a part of a college life. But it is just if it is the once or sometimes it's okay. If it is become your habits, then you are normal. Okay, then you are going to get your mark sheet with the colorful numbers. Okay, it may be, you can say, good to show to your parents or to get laugh on it that, oh my God, whatever I have done, but even though I am able to manage in second class or the first class, you take the proud. But the assessment will come within a 10 minutes, you will get your persons before to you. If it is a knowledgeable guys, generally they are taking the interviewers as a knowledgeable guys, right? Okay. So what they are going to do, they will assess in a way that, wow, you are having the 60%, really nice. In our university, to pass with the 65%, it is a pride. And you may tell that, okay, the 65 you are selling, that is the list. In my class, about 80% students are there. Students saying 80 to 90%, then my expectations will be a lot. 90%? In my university, no one has crossed the 65%. That means either you are very much excellent in your class. Cross boundaries, it is there. Okay. Or else there is a second. Like the questions may be there with you first, or you are able to manage the questions. You are knowing the phenomena, how to handle and manage. Then the selective questions answer you have done. So many possibilities are there, or you may have able to settle in that part, but it was never been going to happen in that way. Okay, so my expectations for the students having the 90%, it will be beyond my boundaries and my imagination. Okay, so I may start, as you are going to consider as a note, you are having the just a level of a bachelor. It's comparatively maybe near to the master's class. Okay, so I may have just some basic questions related to analysis or understanding first, because all the criteria is always there in your particular paper format. Some basic on the informations, some are the analysis. So I may have the some basic few questions based on your subject knowledge. Then may I move to the certain questions, maybe a different. That is based only those who understand that topic are able to reply. I'm asked that I'm looking to isolate the acidophiles having the extreme conditions like a tolerance of you can say the temperature at near about 80 degrees centigrade, and they may able to synthesize specific enzyme. How may I go? Okay, so your answer will be okay. Acidophiles, that is the acidic conditions. pH should be low. Temperature may be a high. Okay, so where I will get such points? So we'll think 80 degree in nature. Well, it's where it may exist. Yeah. Okay, optimum temperature. Where in nature 80 degree, around 80 or 100 degree can be found. Yeah. Hot springs, hot stream, hot springs. Okay, volcanoes, eruptions. Yes, please. Volcano eruptions, such places are there. Okay, so I'm looking for the acidophiles and this. Two phenomena I have covered. Now I'm looking for the certain enzymes to be get synthesized. So what will be my media and culture characteristics? So if you have learned the basic parameters like pH is an essential component, temperature is an essential component, then you may start to apply your mind. You start to give the answers. Otherwise, you may say that acidophile, those who are in acidic and other all, you may start to give the answer will be tumbling. How you are responding in the same questions, is it based on your understanding or it is just based on mug-up? Within few minutes, one able to understand. This is just basic questions I have asked. Okay, that you able to perceive and understand and give the reply. But the same thing I'm asking the different. I may be knowing the name of the one enzyme, those only being produced by the acidophiles. I am just going to target, if you are a 90% guy, you must know that enzyme name. I will ask that I want to get this enzyme. How may I proceed? Okay, so if you are knowing that this enzyme is only available with the acidophile in thermophilic conditions, then it will be related. Then you are going to say the hot stream. So I have not asked the first question directly, the second question I have asked. So those who are having the real knowledge and understanding, they are able to reply. That means you are not a 90% real guy, you are in a 70% falling down. Then I may clue with the clue. You may know such enzymes being produced by some thermophiles. Okay. Then I give one another clue, like acidophiles. If it is yes, then you are in a 70%. If it is not, you are in 60% category. Understood? You are not able to reply in that way also. I have asked about the enzyme, then I moved the thermophile, acidophile, everything is told to you. Then I may move ahead. Okay, that I may give the exact name of that organism. Generally, it is very well known in this place. Okay, and then may I say that have you worked on it? It's like a one E. coli or the some pathogens, Salmonella typhi. A and B is there. We know that they are basic characters. Okay, typhoid, we are knowing. Okay, some bacteria are causing the typhoid, Salmonella typhi. I may ask about A and B, but I have just fever. Such characters are there related to the feveric condition, and it is suspicious for the some bacteria. Can you enumerate? It is maybe because of any, you can say the nematodes or any prokaryotes or any kind of a bacteria. What you are going to respond? It is a prokaryote. Bacteria prokaryotes. Bacteria prokaryotes or eukaryotes? Okay, it's the prokaryotic. Okay, so I have given the one part, whether it's a bacteria. So many things are there. Within the answer, multiple choice questions is coming. The answers could be many. So which one is the best answer over it? Nematode is not going to be there. Never been. But the bacteria and prokaryotes, both the answer will come. Okay. Do you know about the eubacteria, archaebacteria? Okay. So yeah. So many things you may have learned about it. So when it is been asked, you are going to have your own world. Okay. It is not always that a subject oriented. Sometimes it's nearby the questions where it may also happen that within the one questions, maybe you are not able to recall. So 4 or 5 questions have been prepared in a way that to assess you. Maybe you are failing 2, but you may able to give the answer at least 3. Okay. Sometimes happens that at a moment light not yet happen. You are not able to catch the questions or the fundamental things you have learned, but you are not able to recall. But after the 5 questions, if you are coming in the 90% to the 60%, then the chances of you are getting selections because you have done the fair. You are looking that I'm fair in this. Okay, but all it is going to be a unfair things is going on. So this is the real part is coming. Not only for the my chapters or anything or the papers. It may be from the enzyme technology. It may be asked from the other place. Anyway, okay. So many things are there. Let's move ahead into our colony broth characteristics. Yeah. Why need? Yes, already I have told many a time. Why we need to study the culture characteristics? Okay, to understand, to know our culture, whether it is grown in the liquid medium or it is in solid medium. Okay, why we are looking? Maybe it is a beneficial or the harmful organism. I'm doing experimental. Maybe it is in a pure form or in a mixed form. Or maybe I'm working, it is a novel organism or it's a well-known organism. For every sort of things, I need to essentially learn their basic fundamentals. Okay, it is like one child is there, right? And near at home or somewhere. You are after a few days, so they are knowing that child, if it is given the mobile, they are happy. Sometimes not with it. With chocolate is given, child is happy. Sometimes not. Sometimes it is given with a few sweets, child is happy. Sometimes their child is always asking for taking you outer side. So what is the maybe a gender, maybe a matter, as the age is also going to be a parameters with the child. Okay, if it is a 10-year child, then their characteristics may change. If it is a 15-year child now, their characteristics and their requirement is going to change. If it is a 20 years, their requirement is going to change. Right? They are at the child when you are a 5th standard or in a 4th standard, they are happy with one lollipop. Then after, they are just looking for the dairy milk. Okay, then they are not very happy with that also. They are looking for the certain essential different parts. They were earlier maybe happy in the bike. Now they are looking for the other things. So what is the characteristics? According to the age, as the human are changing, microbes are changing. Because I already told, it is our guest or the part to whom we need to cater. It is like a child. So you are knowing the characteristic of your child or yourself or your parents. When to ask, when to not to ask, to mom or dad. Okay, or your friends and colleagues. So how to handle the phenomena or the management is done even with the faculties. You are knowing many things, right? Or you start to predict. So this is the characteristics are there. It is been essentially required to learn. Same thing is there related to culture characteristics. We are looking to understand their physiological conditions. Physiologic, they are the what is their behavioral part. When it is coming for the animals or the human, it is behavior. Here, physiology and behaviors are being connected. Same it is there, we are looking the microbial physiology. So we need to essentially learn their characteristic. Okay, so all the microbes are going to exhibit different kind of a characteristics. So accordingly, we have to manage and cater. Okay. And another motto is what? To classify them. When you are able to learn, okay, our main targeted part is what? To classify them into several groups. If I'm going to have the several activities here, okay, I may give the 5 activities. Your whole group or class is going to divide it into, may in a fifth category or the maybe only one person's may stand, or may in others. I may give the some like a activity, locutions, some the sports activity. Many guys are maybe start. But when I'm going to give the sports, it's not just cricket, it's something else. You may say, no, I don't want. Okay, you may shuffle to another. Okay, so when we want to classify them, we must have certain parameters. For classifying any, whether it is a good or bad, if food is there, you may have some classification. Okay, whether maybe it's veg and non-veg, based on the taste, it is a bitter or the tasty. Tasty means what? It is a sweet or the salty. Sometimes you are like, sometimes you like sour. Why you're taking the lemon juice? Once at a time you are taking the something else. So for the food products even, maybe it is in liquid form or solid form, you have your own criteria. So we are looking to classify every sort of the objects and everything. Okay, same thing is we want to classify our bacteria. Then we need to understand based on the characteristic, but the characteristic is generated based on the certain parameters. So we have to learn their parameters. Okay, so most important parameter, what we are looking, that their growth and reproductions in a liquid and solid medium. Because these are the microbes, they are going to secrete something during their growth stages or in their developmental stage. So that part is very much essential for us to learn, and both their mode of reproduction, because the microbes are growing at the certain stage. When they are going to attain that level, they will start to divide. So if they are just on a growing stage and then reproductive stage is together, then they may not going to give my metabolic essential components. We are going to learn about the growth phases, lag phase, log phase, stationary phase, and decline phase. Such things are coming. Which one? Yeah. The thing is what? That we need to understand the growth and developmental stage. Okay. Growth means what? That means they are going to nourish themselves. Okay. They increase in size, like the child is there. The parents are nurturing like anything for their growth. Okay. They are going to, first it is like a child is there. Maybe in the bacterial cell, if we are looking, the binary divisions are there. Binary fissions we are going to learn. Basic mitosis. In bacteria, mitosis process is not been there, but similar to the mitosis, cell is going to divide. We are going to learn the fission. Okay, reproductive stage in the growth curve. That is in unit number 3. Okay, how the microbes are divided. So once it is divided from the mother cell to daughter cells, okay, the size will be, you can say the bacterial size will be small. So first they will grow. They will get enriched and fully get matured. So during the maturation stage, what they are going to do, they will acquire the material from the outer side. They will release the extra product outer side. Now they are mature. They may look, shall I go for the again reproductive stage or the further metabolic activity stage? Okay, they will wait and halt for a while. If the nutritive media are offered very well, if it is a continuous part, then may go for the reproduction. That means they are going to divide. If it is not there, they are looking for some maturations. It is like getting married after earlier it was I don't know about the cross country wise, but here in India or somewhere we are looking, age is thought like a 20 to 25. It's the average age for getting married. But if you are going in Japan, it may be a 40. If you are going in Canada, it may be same different. Or within the country, a group of the people are get married at 20, some are in 25, some are at 40 or the 45. What could be their motto and agenda? They may think that getting married, get settled, have the happy life. Okay, do your jobs, many things. Some people are career oriented. They will wait. Still, I'm not going to attend the some points. I will not. Okay, so they may wait for a while for this thing. After that, get married. They may have their own relationship that's going on something else, but they are not getting married. Okay, so same like this, the bacteria is doing. What is the human thinking mentality? Bacteria is also doing. Shall I go for the reproductions or shall I get matured enough further? This is the real physiology to understand. Right? You have to relate all the things that is your child or you are as a child and growing stage, you are looking the whole world and phenomena. Same thing is happening with the microbes. If you are going to give the some flourishment, if it is like one guy was a career oriented, right? One person's in the currently in Olympics, he got from the India, right? Got the gold medal. Many things started to get offered. He will be settled in the job, everything. Now he may say that no, I'm going to develop my institute first. I will develop some things, then I will make off. Otherwise, may that okay, now life is settled. Get married, have the some child, have the one family. Okay, he may think in that way because everything is with him now. What is been looked our leaf products are given to them, so they make off the reproduction. If the adverse conditions or not always adverse conditions, but the some facultative conditions are coming, one may go for reproduction stage or else different. So each microbes during their developmental stage, which phenomena are going to move into one to another directions, that part we have to learn. This is the real understanding of them that you can use into your research purpose or to develop any drugs will be there or not. That some pathogens has infected some person. If the drugs is given to them, whether they are going for the development stage or reproductive stage, you are looking to hold which phenomena. If the reproductive stage is coming, then they will start to divide and the development stage, their maturations will go. If already they are matured, so they will start to fight. Okay, only two phenomena. What they are going to do, they are going to release certain chemicals against your medicines is given. So they are fully mature. They are able to fight. Okay, your child when cannot, but they are looking that if I'm only one, I may get died. I will increase my size. Okay, I will increase my population. So few can be able to resist that antibiotic, they can save. Okay, so it is there generally in bacteria. If you are looking, they are copies from the mother to daughter. Okay, much major changes are not taking place in the genetic diversion. It is there, but it is not that much higher. So it is just dividing into two. It is like we are having the so many times some you can say a theories. Dev and Danavs. Okay, so demons are there. If you are going to cut into two, they will get matured. They will survive. Okay, so these are the becomes the demons. So they are looking to, shall I increase my power to fight, or shall I divide into two, get matured, and then I will fight? Or maybe within the two, one may escape from this mechanism, and they will get settled for a while, and it get matured, and then it is going to fight. So such thing is always there. This is the physiology. All the organisms have their own characteristics to do. So when I have kept, in the last week, we have kept our culture in the first in the you can say in rotating condition. Okay, shaking conditions. Same is organism flask where the tubes were containing or the beaker were kept outside. The bacteria come to know that my environment has changed. Some of the tubes only have received the flocculation, some has not. So within the tube, the bacteria were came from the only one place. Source was same, one flask, you all has done the inoculation. But all the tubes having the different response. So you have to note it down each, in which category, which part, the characters they have changed. So they are looking to change their behavior any time. It is like that when you are with your friend, when you are with your parents, same thing is your parent is there and your friend is there. Then you are how responding, right? When your parent is there, your faculty is there. When only your faculty is there, okay, your behavior always change. Only one culture was given, 25 or 28 tubes were being prepared, each having the different characters. Understood? Just two phenomena has changed. Inoculations were done, that was in a static condition, and in a, you can say, in rotating conditions we have kept. Okay, staking or rotating, both are the same. What are the major factors? Yeah. In that conditions, one, it is the aeration that we are going to learn more in detail. But the introductory part, I have opened this part a lot. The reason is that you then after able to understand and like to learn the each points maybe a later, like a pH, temperature, simple things. The seasons are getting changed. Some people like winter a lot. Some people like summer a lot. Within the summer, some people like nights a lot. Okay, some people are nocturnal for a reading. Some people are early birds. Wake up at early morning, they will start to read. So all the people having their own behavior. So one most important will be the temperature for them, then the pH. Okay, then the what are the salt concentrations outer side? What is the osmotality, osmotic pressure outer side? These are the major factors. Okay, other several are also there. Just we are going to focus first in the few early only. Same all are being given detail in the third unit. Okay, why I'm opening this sort of the statement before you? Then you will realize that my bacteria is now at a generally like at a 37 degree. Okay, but all not bacteria. 37 we have kept because our normal body temperature is 37 for a human being. But it is also not a true, it is average. Cross country wise, we are going to take and check the measurement of the temperature, it varies. Okay, some are the very much cool minded, some are the always in a heat conditions or in hot conditions. So all the persons, their blood pressures and the pulse are changed from one to another. So all are okay. Now some for the individual, if you are talking, that is we are just talking that ideal condition is there. For the ideal guys, if we are going to give or change certain parameters, let's start to poke the noise in his life. What he's doing, if he's good in behavior, everything is there. Everything is start to say that you are doing rubbish thing. If he is written everything good in an exam, list mark is given. What will be his behavior? He will start to fight or not. He is the cool-minded. Okay, then you may start to give the some better treatment or the bad treatment. How their behavior change? Because in ideal condition now, we are looking to do this one ideal condition. My bacteria always good in this condition, but now it's changed. Why it's changed? Yes, we are changing. We are the human being. We have our genetic makeup, and the response is there. Expression is coming. Same thing is with bacteria. They are having their own genetic makeup. They are having the expressions systems. Though they are tiny, we are large. But always the with the one parameters to change in the second parameters, there will be a change. So if you have just something in your mind, understanding, then you are able to learn the basic physiology. Then what are being associated? It is like that. By chance, I may poke someone. Okay, in a way that someone has came late, very much late in a class. I may start to shout. He or she may having their own reason. Sometimes it is a fall down at a while taking the steps. I don't know. Maybe having the fevers, he gone for the medicines, or he or she may have missed his bus, but even to continue in a class, he managed or he has managed and came in class. I don't know, but I started to shout. What's going to happen? You or she may accept it sometimes or reject it. Oh, this is the fully rubbish guy. I have managed any things, but he not able to understand what I have done for it. Okay, just to attend the class. Sometimes or some in another way also, I may appreciate. Oh, even the late. I may wait for a reason. And then I have started to give reply. Similar, our objects and everything is changed. Like that, the persons may respond. If I have shouted, he may or she shouted, hormonal changes are starting from the pituitary gland. Okay, till reach the adrenaline gland. Okay, adrenaline or noradrenaline is going to secrete, nothing else. Okay, same phenomena, if he is able to manage, then it's okay. Same thing is happened with this all the kind of microbes. They don't like. If you are going to add the certain things, they will not grow. Why should I grow? Or sometimes very good, you are in a good conditions, everything is there. Food is given without salt, you may not like. Okay, if it is added with higher salt, you may not like. You are going to do the osmotic pressure related questions marked with E. coli. If I'm going to give the higher amount of a salt, what you will do? Okay, so I'm poking the things. Okay, poking the things in their life. Okay, so they may respond in different formats. That means for their life management or their life sustenance, they will start to develop or change their pattern. That means they are going to release certain kind of a enzymes or protein to manage such shock. Without salt, with salt. It is like that salty thing is given, or the for a week, for a month. Sometimes having the so many things is the sawan is going on, right? Fast are doing. Some people are in the rituals. They are having the fast for the holiday for a month. In Ramadan, roles are going on, right? Ramadan is there. So they are how they are managing in different formats. We cannot manage, or we are managing in the some in the different, like Jainism is there. They are having the for a month. They are not going to take any kind of a salt, not much a luxurious, any sort of the elemental part is there, but they are able to manage. So our bacteria will be ready. If it is ready, that means they have changed their behavior. They have started to release certain special compound. Those I am looking to understand. If I'm going to the stringent conditions, then they will start to release certain chemical outer side. That was my motto. It may be useful for when cosmetics, may be useful as a medicine. Okay, so this is the second part is starting, that why we are understanding and study them with different parameters. Drugs means medicines. Drugs means not it is always specific. Drugs in mind is coming the certainly related to narcotics. Not that. Drugs the common term. Some medicine, antibiotics. You can take the one example. If I'm going to give the addition of the certain antibiotics, bacteria will not going to grow. We will learn the antibiotics in more in detail. Mode of action. Mode of actions. That means they are halting the protein synthesis, nucleic acid synthesis, cell wall synthesis, cell membrane synthesis. So many parts are there. Just one antibiotics having the so many controlling areas and agents to be get targeted. I'm just telling about the antibiotics, right? So they may arrest their growth. That means they are not going to may divide. Now they are halted. Okay. Now I may give the certain things which is toxic for them. One drugs, medicines I'm giving, which does not allow the growth of that bacteria. Now I'm going to give the one toxic compound. When they are going to eat it, they are going to die. So what I have done, reproductive stage I have arrested. Okay, what I have done, they are get matured enough. Maturity time is there. I have given some toxic compound, they died. So I have killed them. So both the things are done together. If you are knowing the growth and reproductive stage, all are there. in which conditions this bacteria is going to behave. That is why it is been asked by the doctors how many days you are taking, since long you have taken these medicines, basic history is taken. If this kind of bacteria will be there, I have to treat because they are in the stage, this stage only. Accordingly, the drugs prescriptions will be given to you. For 5 days, you have to take the 3 tablets. Then after, we are going to change your prescription. They're changing, maybe it's a change with a 1 or 2, or they are giving it now only a 1 tablet is required in the morning. So this changes is done based on their growth parameters. So if you have learned these growth parameters, then you can give the pharmacist can give the suggestions to the doctors that you have to do so and so things. Fine. So I'm not going to continue more. Just with the first and second slide, we have ended. Okay, so link is open for you. Those who are in offline mode here can come able to join the lab even, not a problem. Those who are right now online, be in a time. Okay, be in a time. We will first going to join in the class only. Okay, I will tell you certain things before to you what we are going to do in a lab. Then we will move in a lab. Accordingly, we will manage our lunch break. Fine. So put your attendance.